All right, so we are now being recorded. And again, welcome to our sixth video teleconference for BIOL 105 YC. This is our last video teleconference before the midterms week and the midterm exam. Okay, so in this video teleconference, as usual, we'll be looking at the calendar, the schedule ahead, and then we will review cellular respiration and the mitochondrion. Okay, that's the previous chapter. And then lastly, there will be some announcements on what to expect ahead. If you also have questions about photosynthesis and the chloroplast, you may also ask them later on the open forum because unfortunately we won't be able to cover that topic in this review now because of time constraints. So here's our schedule. We are on time with video teleconferences. In fact, we are on our sixth instead of the fifth. And the topic on photosynthesis and the chloroplast should be wrapping up today. I did not post yet the group discussion, but that's fine. The group discussion is still for chapter 5, so I hope you've accomplished that one already. Um, the midterms week will be next week, but as I announced last time, our midterm exam will be opened on the last week of February. For your section, I think it's gonna be February 27. Okay, so you'll have enough time to study for the midterms. And these are the chapters that we've covered so far. So I believe we are halfway of the chapters that we need to cover for this semester. And this morning we are going to review chapter 5, aerobic respiration and the mitochondrion. So I hope we can have a discussion about it. No, I will be asking you questions, and I hope you'll be able to answer as well and share your thoughts on this chapter. Okay, so we have several things to talk about on this chapter, so we'll start right away. Um, this is going to be a bit of a complicated chapter, especially if you really read the book. And I really recommend that you read the book than just view the videos. Um, for many of you, perhaps, if it was a long time ago when you had a lecture on cellular respiration and the mitochondrion. So, before we delve into that particular topic, let's look at this overview of metabolism first. So, metabolism is the total chemical reactions that happen inside the living cell. Now, so there are two types of metabolism. We can categorize it into two: anabolism, shown here towards the left direction, no? Also in blue arrows. And then we have catabolism, shown here towards the right direction, the green arrows. Notice also that we encircled here what happens to ATP in both anabolism and catabolism. So can anyone in the group shortly describe what anabolism or catabolism are? Based on the illustration here.
Um, anabolism, sir, is the making up of macromolecules from its what do you call this, from its building blocks. While catabolism is the total opposite, so it's the breaking down of the macromolecules into its building blocks. And furthermore, to from the diagram, for carbon dioxide, water, and the ammonia.
All right. So you are correct. No, that's correct. So the breaking down from macromolecules. So your keywords were macromolecules or the biomolecules, and then the monomers. That's what you meant by their building blocks. So you are correct with that. No. Um, when we eat food, now if you had your breakfast, We eat food for the energy that's in food, the energy and the nutrients. Why? Because our body, especially our digestive system, will be able to break down food physically and chemically into its constituent molecules. And the cells in our intestines will be able to absorb those nutrients, no, into our bodies and distributed to our cells. And then in our cells, furthermore, those nutrients are going to be broken down. No? So from lipids, polysaccharides, and proteins, they will be broken down into their monomers, like the amino acids and the sugars, or the building blocks of the lipids, like triglycerides and the fatty acids and the glycerol molecule, and they will be used by the cell, no, for energy. So that's catabolism, the breaking down. Sometimes if you starve yourself, your body will use your own biomolecules, no, to break down. So that's the reason why if you starve yourself, no, you will grow thinner and thinner because your body uses the proteins in your muscles as a source of energy. No, that's catabolism. The opposite is anabolism, the building up, no, as you mentioned. So we break down those molecules, but then we actually use the building blocks again to build our own bodies, our own cells, and that's anabolism. And the result will be the biomolecules. No, you pointed out that the waste product of catabolism are shown here as well. So we have carbon dioxide. No, we exhale carbon dioxide. There's also water and ammonia. So carbon dioxide and water, you can actually observe that in your exhaled breath. No, if you have a glass in front of you and you sort of breathe on the glass, you will see condensations. Those are actually the water that are condensing. And in your urine, that's where you release the ammonia. All right. So let me continue. Um, if you're also familiar with the anabolic steroids, no, used by bodybuilders, no, that's actually an application of anabolism, the building up of our own bodies, no, our own cells, to mean. Um, you also see here, very important to notice, what happens to ATP. No, when in catabolism, when the nutrients are broken down into their constituents, the bonds between them are broken, and the energy in the bonds are transferred to produce ATP. So that's the one in the bottom here, no, the one in the bottom. So we produce ATP through the breaking down of the nutrients. And the opposite is true when we try to build up our bodies. We use ATP and ATP becomes ADP. All right. So are there any questions about anabolism and catabolism or additional information before we move on to the next topic? For those who just arrived, we were talking about anabolism and catabolism. Okay. The illustration here, this is just a simplified version of metabolism. In reality, it's really much more complicated than the maze that you see here. No, it's a lot more complicated. Now, in this particular chapter, we are going to focus on cellular respiration, as you see here in the middle. So this one, if you can remember, pyruvate, acetyl coenzyme A, TCA cycle. So this is where we're gonna focus in our review this morning. And that's catabolism, of course, catabolism. So our topic on cellular respiration might be very far-fetched from our previous topic, no, the cell membrane, because this is about energy production in the cell, but in reality, they're actually much more related than you have been thinking, perhaps, no? Because one function of the cell membrane or the membranes in the cell is also for energy transduction. Now, as you see here, number 7, this is the mitochondrion, which functions as the energy powerhouse of the cell. No, um, so essentially, we're still talking about one of the functions of the cell membrane here. And last week we were talking about solute transport as a function. So now energy transduction. So here is a visual representation of the important processes that happen in cellular respiration. No? We have oxidation and reduction. So you see here we have several molecules from methane, which is the most reduced, and then it's slowly being transformed until it becomes carbon dioxide, which is the most oxidized. Okay, can anyone from the group um, tell us what do you think is the meaning of the color gradient in the arrow here? Why is it redder in the most reduced state and then bluer in the most oxidized state? What do you think is the meaning of that?
From the most reduced state, sir, it's red because the carbon it's more electronegative than the hydrogen. While going through the process, since it binds to oxygen, which is more electronegative than carbon, so it attracts the electrons more than since in the carbon dioxide there are two oxygen, so all the 8 electrons are attracted to the oxygen.
Yes, you explained the mechanism there perfectly, no? Okay, so that's correct. Everything you've said is correct. But let me ask a question based on what you said. What will be the consequence of that? No, everything that you said about the electrons, the electronegativity are correct. But what's the consequence? What will we expect if the substance like methane is reduced versus a substance carbon dioxide is very much oxidized? What will be the expectation or the difference?
I think it's the reduced state is hard to break than the oxidized. I'm not sure.
I think you got it
Uh,
reversed.
I reverse. Okay. The oxidized states they are hard to break than the reduced state.
That is correct, no? And when we break bonds, what do we release? You said most reduced, easier to break the bonds. You're correct. And when we break the bonds, what do we release? Anyone from the group?
We release energy, sir, in the form of heat, sir.
Yes. That's why methane, if you remember, methane is combustible. It's a fuel, di ba? You can burn it because the bonds, as you mentioned, are easier to break because it's very reduced. But good luck burning carbon dioxide. Have you seen someone burning carbon dioxide? It's impossible because you cannot anymore release the energy in the bonds of carbon dioxide because the oxygen, as you mentioned a while ago, are already holding on to the electrons really, really tightly. So you cannot break the bond. But for methane and for related substances like methane, like butane, gasoline, petroleum, those are all in their most reduced state and it's easier to break the bonds and therefore they can be used as fuel. Okay? How do we relate that to the cell's source of fuel or energy? We have here two types at the bottom. Here on the left, we have fats, triglycerides. And here on the right, we have carbohydrates. We have starch and glycogen. Now, and here on the far right, we have a monosaccharide. So, how is the concept of reduced state related to these sources of energy in the cell. Look at the structure of the macromolecules at the bottom. Why is it that the cell uses fats and carbohydrates as sources of fuel? Why not proteins? Proteins may be used, but why is it that the cell considers fats and carbs as the main sources of energy? Why not proteins? Why not nucleic acids? All right, let me ask another question. All right, we know that these are sources of energy for the cell. Which among the two, lipids or carbohydrates, are more energy rich?
The fat, sir.
Yes, not the fats or the lipids. Why?
Because sir, the basing on the triglyceride structure, it is made up of the majority of hydrocarbons, which we can see are attached to the glycerol backbone. So fats are in most reduced state than carbohydrates. So therefore, fats have more bonds that may be broken to provide energy for the cells.
That is correct. Okay, that is exactly correct. So let me just point out for the carbohydrates, you can see here in the monomer here on the right, no, that there are OHs attached to the carbon. So that's sort of similar to the formic acid that we see here or the methanol that we see here. So it's although it's energy rich, it's not as energy rich as the triglyceride that we see here, wherein all of the atoms attached to the carbon are actually hydrogens, no? Like how methane is. So that's correct. However, for the cell mainly uses fats as storage for energy. And instead, it decides to use glucose, a type of carbohydrate, as the fast and main source of energy, for the cell. So there's that choice made by the cell. So our fastest source of energy will be the sugar, glucose. And that's what we're gonna talk about in cellular respiration. But first, let's focus on glycolysis. Glycolysis is strictly not cellular respiration. Okay, it's different. However, it produces a product that will be used later on in cellular respiration. So here we can see there are 10 steps in glycolysis. Each of the step is catalyzed by an enzyme. No? So you look at the names here of the enzymes. Now, let me tell you in advance, you don't need to memorize all of these names. What is needed is for you to understand what generally is happening in glycolysis. No? So we start with glucose, a molecule of glucose. And then there is this phase of glycolysis we call the energy investment phase. Why is it called energy investment?
Because it uses ATP, sir, in the process to
Yes,
eat.
that is correct. Instead of producing ATP, it uses ATP. No? And the reason really there is to phosphorylate, these portions of the sugar molecule, which causes the sugar molecule to split. So that's the reason. It will now split into two, and these two will be furthermore processed into the second phase of glycolysis we now call as the energy payoff phase. Why energy payoff?
Because it uses ADP to form ATP, sir.
All right, so ATP is now formed from ADP in the second half of glycolysis. Okay, so as you see here. Now, the final product of these 10 stages of glycolysis will be a 3-carbon compound, two 3-carbon compounds because glucose is 6 carbon. So the product will be two 3-carbon compounds we call what?
The pyruvate, sir.
All right, pyruvate. So you see, you have a net product of two ATPs. Okay, there's two ATPs net product. We have two pyruvates. And lastly, shown here will be this NADH. No? The NADH is an electron carrier and it carries electrons which will be later on used in the citric acid cycle in cellular respiration. So those are the important products of glycolysis. So please remember those. Lastly, where in the cell does glycolysis happen?
In the cytoplasm, sir.
All right. So glycolysis doesn't require a special organelle for it to happen. It can happen in the cytoplasm. That's the reason why even prokaryotic cells can do glycolysis, no? Because they don't have a mitochondrion, but glycolysis will happen in the cytoplasm instead. Okay, so this is just an illustration of how glycolysis slowly burns the glucose, no, slowly. So with the help of enzymes, our cells are able to burn the glucose molecule into a state which has lesser energy. And in the process, it doesn't reach a temperature that's very hot. Otherwise, it will die, no? Thanks to the enzymes. Remember, there were 10 enzymes involved. Um, if you don't use enzymes, if you just burn glucose as shown here, you would still be able to release the energy from the glucose, but once released, it's lost in the air. It's useless. So in glycolysis, we release the energy from glucose and turn it into ATP. And then we get the electrons and give it to NAD plus. No? Okay. Speaking of NAD plus, here are the structures of similar molecules to NAD plus. No? So here on the left, we have NAD plus. Once it gains an electron, as you see here, it becomes NADH. And here's a version that's found only during photosynthesis in plants. So we can forget about NADPH for now. Question, what does NAD stand for? The meanings are here. Oh. So what's this? What's the first one? N.
Nicotinamide adenine dinucleotide.
All right. Nicotinamide. No, nicotinamide, adenine, dinucleotide. So meaning to say it's like a nucleic acid. So look at this structure. This is actually ribose, oh, like the sugar in your RNA, no? So and then there are two of them attached together with a phosphate backbone. So it really looks like a nucleic acid nucleotide. So that's why it's called nicotinamide adenine dinucleotide. And it's a carrier of electrons. So as you see here, the electron is stored here in this nicotinamide portion. No? So look at the difference. From here, there's a double bond. Once an electron is carried, the double bond becomes a single bond, and the electron is used to carry the hydrogen atom there. No? In plants, the NADP stands for nicotinamide adenine dinucleotide phosphate. Okay, but that's not related to our cellular respiration story. So let's think about NADH, the electron carrier, okay? So remember, NADH is one of the products of glycolysis, and it's used to carry electrons. So it needs to carry electrons from this portion to another portion. So where could that be? So we'll see later on. Now, before we go to the process of cellular respiration, let's figure out first the problem of glycolysis. And what is that? What is the problem of glycolysis?
How to produce more NAD, sir?
Yes. Okay. So before you continue, this ATP here is gonna be used by the cell for energy, di ba? For activity, and after using it, it's gonna become ADP again. So no problem with ATP. No? Pyruvate, of course, is a byproduct of glycolysis, but when it accumulates in the cell, there's really no problem. Okay, it's not a poison. But the problem is, in order for glycolysis to continue, there needs to be an electron carrier in the form of NAD+. But the problem is, they're gonna be used up to become NADH. So, in the long run, the cell will run out of NAD+. So, question, how will it renew its NAD+ source? How will it produce more NAD pluses?
NADH, sir, it's gonna go to the pyruvate and it's gonna produce lactate, and producing another NAD, sir.
All right. So the solution here is NADH is gonna give its electron to pyruvate, the byproduct, and pyruvate will become lactate or lactic acid, no? And NADH now becomes NAD plus and it can go back again to the glycolysis, no? So this is how the cell solves that problem, no? And usually the cells that have this problem are those that do not do cellular respiration. Those that mainly rely on glycolysis, like many of the prokaryotes, no? Many of the prokaryotes can can only do glycolysis. And look at the product here. This is lactic acid. This is the same product found in your yogurt, for example. So what is this process called? This whole process?
Lactate acid fermentation, sir.
Yes, so this is the lactic acid fermentation, no? Used in the production of yogurt, many other food products, di ba? Can be produced, can be enhanced in flavor using lactic acid fermentation. And then there's another form of fermentation wherein pyruvate is first transformed into acetaldehyde before NADH can give its electron, and acetaldehyde becomes ethanol, no, and NAD plus is renewed. But then ethanol is an alcohol that we can say is edible, and we also use this process to produce alcohol, no, alcoholic beverages. And this is also fermentation, but because it produces alcohol, we call this alcohol fermentation. Okay, so that's fermentation. It's actually just glycolysis and then NADH giving its electron to pyruvate, not to produce a waste product. So that's glycolysis. Any questions so far about glycolysis? Okay, there's none. Let's move on. For cells that have mitochondria, of course, these are eukaryotic cells. They can, of course, perform cellular respiration, no? Not just glycolysis. Okay, so this is a structure of the mitochondrion. You should be familiar with this. The mitochondrion has two membranes. There's an outer membrane, no? The thin blue line that you see here, no? That's the outer membrane. There's an inner membrane, also the thin blue line on the inner side. And between the outer and the inner membrane, in orange here, will be the intermembrane space. And on the innermost portion of the mitochondrion, the yellow-orange color that you see here is the matrix of the mitochondrion. And the inner membrane, as you can see here, is also folded, no? Folded inwardly to increase the surface area. And those foldings are known as the cristae. Okay, the cristae. So it's important to be familiar with this so that we will know where the certain processes of cellular respiration happens. Okay, so this is an overview of cellular respiration. Now, you actually have cells in your body that can switch between glycolysis only or cellular respiration. An example of this is your muscle cell, no? Your skeletal muscles. If you exert a lot of effort when you do exercise, no? Especially if you don't do exercise often and then you do a lot of exercise right away, your muscle cells will cope with your strenuous activity by just doing glycolysis and lactic acid fermentation, thereby producing lactic acid. And that's what we feel in our muscles as pamaol, di ba? So muscle pain. And then it will go away if you rest your muscles, no? But your muscle cells, because they contain mitochondria, they are also capable of doing cellular respiration. So as we see here, the pyruvate produced in the glycolysis can actually enter the mitochondrion, no? And the NADH, which carries the electron from glycolysis, will be able to pass the electron inside the mitochondrion, no? And the ATP from the glycolysis will contribute to the production of, to the additional number of ATP that's produced in cellular respiration. So now, let's focus on cellular respiration. Starting with, how does NADH pass the electrons into the mitochondrion? The catch is NADH from the glycolysis process cannot enter the mitochondrion. So it needs to pass its electron to certain other carriers. As we see here, first it passes its electron to DAP, no, or dihydroxyacetone phosphate. And then in the process, DAP becomes glycerol 3-phosphate. And glycerol 3-phosphate enters the outer membrane of the mitochondrion. And it cannot enter the inner membrane, so it will pass the electron again to G3-PDH, or glycerol 3-phosphate dehydrogenase. And glycerol 3-phosphate dehydrogenase passes the electron to FAD, or flavin adenine dinucleotide, and in the process that becomes FADH2. While your glycerol 3-phosphate returns back into becoming DAP, and it waits again in the outer membrane for more NADHs, no? So this is what we call as the glycerol phosphate shuttle. And the main purpose of this is mainly to get the electron from the NADH from the glycolysis so that it can enter the mitochondrion. It's a bit of a complicated thing, no? Just to pass electrons. Okay, but once the electrons are inside and the pyruvates are also inside already, we can then go into the first process that contributes to cellular respiration. This is the TCA cycle, or the tricarboxylic acid cycle, or you may also call it as the citric acid cycle, no, or the Krebs cycle. So as you can see here in step 1, pyruvate enters the mitochondrion. Okay, but before I continue, just like in glycolysis, you don't need to memorize each of the steps here. No, there are 9 steps here, but you don't need to memorize all the names. But I will mention what are the important things to remember. First, pyruvate. Pyruvate needs to enter the mitochondrion, and once it enters, it's gonna be bound to acetyl to coenzyme A. No, you have coenzyme A bound to pyruvate. And in the process, one carbon is released from the pyruvate as carbon dioxide. So carbon dioxide is now released. And the binding of pyruvate with coenzyme A produces acetyl coenzyme A, which now participates in the tricarboxylic acid cycle. Okay, how? First, it's bound to oxaloacetate. That's number 4. And once it's bound to oxaloacetate, it produces the first product, citrate. So 5 things to remember. 5 things. First, you have pyruvate, and then pyruvate is gonna be bound to coenzyme A to produce acetyl coenzyme A. In the process, releasing one carbon as carbon dioxide. And then acetyl coenzyme A will be bound to oxaloacetate, thereby producing citrate. Okay, those are important stuff. Now, citrate will now go in the cycle, no? And the point here in the cycle is really to strip your pyruvate of whatever energy that's remaining in it, no? As you can see here, carbon dioxide is released here and there. That's an important step, no? And not only that, the most important product in TCA is what? Can anyone tell us what's the most important product in TCA? Is it carbon dioxide?
NADH, sir.
Yes, NADH, the electron carrier. Yes. And is there another one?
FADH.
FADH2, another electron carrier. They are the most important products here if ever we want cellular respiration to continue, no? But aside from that, we have other products as well. Now we have GTP, which is an energy source like ATP, but we will not talk about it right now. And then we have the waste products, carbon dioxide. So those are the waste products. But the most important product will be the electron carriers, NADH and FADH2. So now that we have produced those two, we can now proceed to the electron transport chain. So we see here how NADH and FADH2 will actually give its electrons into the electron transport chain. Okay, but before that, if glycolysis happens in the cytoplasm, where does the TCA cycle happen? You look at this illustration. Where are the NADH and FADH2 from?
Sa Christie, sir.
No, the cristae is on this side. Where does this happen? This is actually from the TCA.
In the matrix of the mitochondrion, sir.
All right, in the matrix of the mitochondrion. So these are fresh from TCA, guys. No, fresh from TCA. So they are carrying with them electrons. But before we proceed, I have a very interesting question. No, also for myself. Sir, you said NADH and FADH do carry electrons. But why is it that they have hydrogen instead of electrons? Can anyone answer? If you can answer that one, then you're good. Why is it that they have hydrogen instead of electrons? And then here they're giving away electrons. Why must hydrogen be present there?
Because they are in their most reduced state, sir. Because as mentioned earlier, the more hydrogens a molecule has, the more electronegative it is, sir.
Okay, all right. They are in their most reduced state. You are correct. No? Sige. So maybe we can start with that. You're correct there, but let me explain. The reason why hydrogen is also present here is because NAD plus and FAD plus cannot carry electrons by themselves or electrons alone. No? You cannot carry, for example, if you're NAD, you cannot carry an electron na electron lang, ganyan lang. Di yan pwede. If you're carrying an electron, you have to sort of carry what's with the electron, and that's the hydrogen. No? So, um, if you don't carry the hydrogen, you will become a charged ion, no? Because you're carrying an electron. So you have to carry with it the hydrogen. So I hope that explains things, no? So in fact, in the illustration, you can see the electron here. Let me erase these. You can see the electron as the orange circles. Now, those are the electrons. The hydrogens are mainly there to help the molecule become stable. Okay, so now that we sort of understand that, the NADH and FADH2 will be able to give their electron into the electron transport chain. So both of them. So there is this chain. This electron transport chain is found in what membrane is this? The one in the middle. Is this the outer or the inner membrane of the mitochondrion?
The inner membrane, sir.
Okay, that's the inner membrane. So if we're gonna go back to the illustration of the mitochondrion here, I cannot expand this, but can you imagine from the yellow, orange to the orange portion? That's the direction, no, of the electron transport chain. Okay, so that's what is being shown in this illustration. Okay, so this is the inner membrane. So NADH and FADH2 will pass their electrons. And since they gave away their electron, what will happen to the hydrogen that they are holding?
It will be released, sir, as protons, sir.
Yes, so if you erase these electrons, no, that I have here, this hydrogen is gonna be released as well because nawala na yung electron used for the bonds. So they will be released as protons. And as you can see here, as the electrons are being passed from one complex, because this is known as a complex, no, protein complex, to another, the protons are released and they are also being pumped from the matrix to the cristae, or another name for this will be the intermembrane space. Intermembrane space. Okay, so that's what's happening to the protons that were being carried by the NADH and FADH2. Okay, and so with the other protons. So before we go to that story about the protons, who will be the ultimate acceptor of the electrons in the electron transport chain?
An oxygen, sir.
All right, the oxygen gas, no, the oxygen gas. So oxygen receives the electron, as you can see here, and of course, hydrogen will be part of the package. And so oxygen becomes water in the process. So water here is a waste product. So it's a waste product. So another question, where were the electrons from in the first place? Now, before they were in the NADH and FADH2, saan galing ang mga electrons? Let's just answer that first. Where were the electrons from? Saan galing ang electrons? Which molecule did you break so that you can remove the bonds and the electrons in the previous processes?
From pyruvate, sir.
From pyruvate, and pyruvate was from?
From glucose, but sir.
Yes. So if you can remember, in the glycolysis, glucose was broken into two pyruvates. Remember, meron ring NADH, di ba? Because the broken bonds, the electrons have been transferred to NADH. And then pyruvate entered the TCA cycle. It was broken some more. Carbon dioxides were released here and there. And more NADH and FADH2 were made because they were getting all those electrons from the broken molecules of pyruvate, which is from the broken molecule of the glucose. And now here are the electrons being passed to the electron transport chain. And the final acceptor is oxygen to become water, a waste product. Okay? So I hope you are able to visualize that one, okay, if you haven't yet. But let's go to the other purpose of this electron transport chain, and that's the movement of the protons from the matrix to the intermembrane space or the cristae. Okay. Question. Why do we need to move protons from the matrix to the intermembrane space? Why is it important to have more protons here than on the left side? The answer is still in the illustration.
To increase its gradient, sir, to increase electron energy and thus producing ATP in the long process.
All right, so when protons are pumped from the matrix to the intermembrane space, you're actually producing a proton gradient. Now, it's like storing water on one side of the dam. Now, we're back to that story again. So now that you have enough protons on one side, okay, you may then slowly release them back to the matrix. So as you can see in step two, from the intermembrane space, the protons are released back into the matrix through this structure. What is this structure called? It's like the turbine in a dam. When water rushes to the other side, the turbine will move and produce electricity. So this time, this one will move also like a turbine and produce ATP. What is this structure called? ATP, because it makes ATP. ATP syn.
ATP synthase, sir.
All right, this is the ATP synthase. And it's where the protons pass through from the intermembrane space back to the matrix. And the ATP synthase works like a turbine and produces ATP from ADP. Okay, so these two processes need to happen hand in hand or together, no, side by side. So first we have the electron transport chain here in step 1. And what is step 2 called? Step 1 is electron transport chain. What is step 2 called? Acronyms O and P. What does O stand for? And what does P stand for?
On, sir, oxidative phosphorylation, sir.
All right, oxidative phosphorylation. Okay, oxidative because you can see in step 1, oxygen is the final electron acceptor, di ba? Okay. And then phosphorylation, what are we phosphorylating here? Where are we adding phosphates here? In step 2. Tingnan nyo, from ADP, naging ATP. How did that happen? By adding one phosphate group. This is where the phosphorylation happens, guys. From ADP, naging ATP. Okay? So, every time you add a phosphate to an atom or a molecule, you are doing phosphorylation. And in this case, ADP is phosphorylated to become ATP. And what type of phosphorylation? Oxidative phosphorylation. No? And that's only possible thanks to step 1, the electron transport chain. Okay, so I believe this will be our last slide. We will not discuss anymore in detail these other ones. So this one shows the electron transport chain. I discussed this in the recorded video, and you may also read about it from the book. So because of time constraints, although I want to talk about this some more. And here we have the structure of the ATP synthase. Look at it. It looks like a turbine, no? And it really, the middle part, the gamma subunit, no, the one here looks like an axle, is the one that turns every time a proton is moved, no, from the matrix to the intermembrane space. Okay, so you can see here how it's shaped like a wheel, and then here we have the axle of the wheel, which turns. We cannot. Here's a mechanism showing how the proton enters and then moves the axle so that it will turn. And here's a mechanism on how ATP is produced from ADP. Okay, so I'll leave those details for you to study na lang siguro. No? Just few reminders, the quiz, if you haven't taken it yet, it's still open until today. I think it will be open until late afternoon, but warning, e-learn will be under maintenance late afternoon until 12 midnight, I guess, today. Um, Group discussion for chapter 5 is already posted. So in your own time, please accomplish that one if you haven't yet. Okay? And the midterm exam will be on the last week of February, particularly on the 27th. The coverage will be chapters 3, 4, 5, and 6. Now, chapter 6 is all about photosynthesis. So I hope you've read on that chapter, and I hope my videos were able to give you more understanding on certain stuff. Because we are running out of time for questions on photosynthesis. Unless, do you have a class after this? Baka meron pang ibang questions. Don't worry, I will not extend 1 more hour or 10 minutes. I will not do that. I'm just asking, do you have a class after this?
Wala ra, sir.
Wala lang. So do you have a question about photosynthesis though? Let's just talk about it generally because I did not prepare a presentation for that here. What is accomplished in the light reaction? What is split during light reaction? It's okay if you cannot answer. um, that's actually found in the videos, no? So please view the videos as well. Read your book, okay, so that you'll be able to distinguish between the light reactions and the dark reactions of photosynthesis and the products of each of those, no? The ultimate goal of photosynthesis is to produce food in the form of sugars. That's the complete opposite of cellular respiration, which is the breakdown of sugar. Okay. But anyway, other questions, not necessarily about photosynthesis. Concerns, questions, clarifications?
Sir, what will be the type of exam for the midterms?
All multiple choice items with one answer per item. 50 items at least.
Okay, sir. Thank you.
So please study on photosynthesis. We might be able to review about it pero after the exam na. And anyway, I think I still owe you a quiz on photosynthesis, and the quiz, this is weird, the quiz on photosynthesis could happen after the exam, which also contains photosynthesis. So we have to deal with it because our time scheduling is quite, um, we also need to adjust now in terms of time scheduling. So we will review in photosynthesis, but after the midterms. Other questions, concerns? All right. Just a reminder though, because this is a midterm exam, I will be opening a platform there in eLearn for you to upload no, a picture of your admit to exam permit. Maybe mga next week na siguro, no? I will just inform you, I will message you in eLearn that it's already ready for you to post your admit to exam permit pictures as a basis. But I am thinking that even if you don't have an admit to exam, you can still take the exam, but I will not be releasing your grade for the midterm. So I think that's a good enough deal for that. Um, if you have more questions, you can message me in eLearn or message me through Messenger and Facebook. Okay, so since we are now over time by 4 minutes, I have to dismiss you. So thank you very much, guys, for joining me in this video teleconference. A recording of this will be posted later on in eLearn. And I hope you'll have a productive day ahead. If you haven't taken the quiz yet, please do so as soon as possible. And again, thank you very much for joining, and you are now all dismissed.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.